Okay, so we'll get started. Thank you for submitting your assignments and not asking for an extension. This is too bright, right? Okay, so I thought I didn't do a good job of both actually motivating the problem of alignment and also probably rushing through some of the things, even though we tried to take it slow last time. But in terms of the local alignment stuff, I think there were a lot of gaps in the knowledge. So before we begin our lecture formally, we will finish talking about alignment. We'll cover that in slightly more intuitive details and just see what is it all about, right? So the central goal behind doing alignment is you have two sequences and you want a measure of how similar these sequences are. And the reason you want this is because you want access to the information that what overall are these even similar or not. And one way you could do this is by taking those sequences, figuring out where exactly in my if this is my original sequence, where does it happen to come up from, right? And this question you can ask for a variety of reasons. One, of course, is, for example, if I do my own DNA sequencing, if you do your own DNA sequencing, are those similar or not? The first entry point, of course, is somebody needs to give you that sequence. But once you get that sequence, to compare, to figure out whether what is similar and what is different requires that step of alignment. And that's the central goal behind both the algorithms, which we're going to talk about in this class, which is global alignment and local alignment. We can use brute force and we can start from this sequence and try to figure out where exactly does it align. But as you saw in the previous class, and you'll see this again in your assignment, these number of alignments are exponential or astronomical, in other words, which means that even for very short sequences, if you were to actually do alignment, it's going to take very, very long by brute force approach. And that's where the dynamic programming approach comes handy, and that's what the two algorithms are all about. So global alignment is broadly a graph alignment problem, right? So what this graph represents, the row represents one sequence x1 to xm, and the columns represent the other sequence y1 to yn. And I am essentially looking for what is the possible. I am not allowed to permute. I cannot change the individual bases. I'm not allowed to flip. I'm not allowed to do all that stuff. If I'm not allowed to do that, all that kind of stuff, what I'm looking for essentially is what possible alignment should I get so that these two sequences can be aligned tail to tail. It doesn't matter whether m is equal to n. It doesn't matter whether m is very, very small. The goal here is to start from the node at the top. The node at the top is where your alignment begins. This is the start node, and all the alignments in global alignment will always start with two dashes. Those two dashes mean that there is nothing there to begin with, right? That's how you initiate an alignment. And as you add each single nucleotide to your graph, as you traverse this graph in one way or the other, and we'll see very quickly how do we traverse this graph, we keep track of where did we come from. And remember this phrase about where did we come from, because it will feature in the actual lecture today, because it has a very special meaning, right? So where did you come from? In this case, you can only come from three possible paths. You only move forward in life, right? So you only you should move forward in life. So for each base that you see here, or each node that you see here, there will be only three possible paths that you can come. You can either do a deletion in sequence Y, you can do a deletion in sequence X, or you can force align the two bases, which is match or a mismatch. The diagonal represents that a base aligns to base. The other two directions, which are perpendicular, represent that there is one indel, either in sequence X or sequence Y. And that is the only three possibility that you have for arriving at a particular node. So we defined this score. Why are we doing this, right? The reason we are doing this is because we are looking for the best possible way to align two sequences, and we want to be able to do this quantitatively, and that's what the central goal here is. And for that, we defined this dynamic programming approach, or we took this dynamic programming approach that was developed in 1970s with this particular aim in mind, that you have two sequences, you want to be able to put them next to each other. And while this is developed for sequences, there are other applications, of course, of the same algorithm. You can think of this as also, if you have very noisy signal data, uh, could be sound signal, could be image signal. If I gave you, for example, this is song one, and its modulation looks like whatever, something like this, where each of these bases have some special frequency. I could give you some other signal, and I could ask for what is the correspondence for one particular portion of the sound. That is an alignment problem. There are other ways to solve this problem in the sound domain. One particular algorithm is called dynamic time warping, but you could in principle also use, I mean, the central principle is of dynamic programming basically there, which is you keep track of each problem at each step. You're trying to maximize the score that you get. Essentially, that's what the dynamic programming algorithm is trying to do here. So for each node, I take the maximum of where I was previously, and I add the score for that corresponding step. That's essentially what your scoring matrix tries to do. In this case, you have to, every dynamic programming solution requires an initialization. The initialization here is a function of what you want. What do I want? I want both of these sequences to align at whatever cost possible. So I want x1 and xm and yn to yn. Once I've done the alignment, I should be able to simply put the entire sequence in one box, which means that it's an end-to-end alignment. The only possible way to do this, well, one sort of approach which will allow you to do this is, the reason we don't prefer gaps, right? So whenever we align a base to this, or we align something to t, generally in biology, deletions are not good, or they're not very preferred, because we know that if the deletion happens, for example, in a particular let's say if this is a start codon, and if this deletion happens here, it changes the entire meaning of what this codon is. What is a codon? A codon codes for protein. One codon essentially results in one amino acid. So if this is going to make some codon M, I don't remember what this makes, but let's say this makes L, the entire context changes because of this deletion. Whereas if you allow for substitutions, or if you allow for matches, those are still okay, because overall it's not as catastrophic as having a deletion. This is called a frame shift. Because of one mutation, your frame will get shifted. We will talk about it later in the class. But that is the premise of why mutations often are very hard, or very sort of not very preferred overall in this setting. So the way we initialize our nodes is by this simple approach, that if we are forcing things to align to a dash, which means a deletion, deletions are not preferable, so that's why they have a negative score, and we start from -1, -2, because we are forcing, in this case, all these bases are going to get aligned to one deleted sequence. So of course you don't want the first row ever to sort of be utilized at all. Same thing for the first column as well. That's how the initialization happens, right? So in this case, I'm initializing so that everything gets aligned to a dash dash dash. This represents that what everything gets aligned to to a dash 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 on this side. Once you have done the initialization, you start proceeding, and you have to still fill in the entire matrix actually, right? Like that's the only way to figure out. There is no shortcut. There are no shortcuts in life. There are no shortcuts on that path as well. That shortcut, essentially, the only shortcut that you can take is to go through these three possibilities for each node. So for this node, I can come from here, which means that I can either do a substitution or an alignment based, or a match based on what sequence exists here, or I could do a deletion in this direction, or I could do a deletion in this direction. So it's 0 if I allow for, depending on what my math score is, and I'm not sure, I should have shown the math score here, but uh, actually it's minus 1, 0 for a mismatch. This is for indel, and this is for match. I will add this one. Once I put the slides up. I had put the slides up at some point. Is it there? Oh, it's there. Okay. I couldn't see the left side. Sorry. Okay. Thank you. Okay. So that's what the score is. Good. I don't have to update anything. That's great. Any questions about this? Any? Yeah. So we'll come to it in slightly later part of the class, because the reason the substitutions are favorable is what we have seen previously. If you took multiple different DNA sequences and you try to put them together, and let's say that was done through some black box algorithm, what you notice is while substitutions you see more frequently, deletions are not as common. The reason for that is also because in most cases, these substitutions will also be at sites which are not going to necessarily change the protein. You can have a substitution without actually changing the protein. But the moment you do deletion, the chance of having a different protein actually increases, right? So that is one of the reasons you prefer substitutions over something. We had discussed this, right? There are basically broadly two classes of substitutions that happen: transitions and transversions. Transitions are more common as compared to transversions, which is something we learned from biology, right? So the scoring is something that has come from biology. So that's why substitution is okay to get a penalty here. Okay. So once we have this, of course, you will not. You will never ever keep all the possibilities, because all you retain is where you came from, right? Where you came from. That is what you keep. So for this one, I know I came from here. Then this one, I could have either come from here or I could have come from here. Why? Because they're both resulting in the same score. So I will keep both the arrows. Then once you go down somewhere here, it can happen at multiple places that any two choices, right? So two roads diverge in the yellow wood, and you can take any one of them and it doesn't make any difference. So that's what this particular um, path shows, that you could have reached here from any of these two nodes. Similarly, this one you could have reached either by doing a substitution or by doing a deletion. So it's a function of what your scoring matrix is. So that's why if this changes, this will also get updated. What will happen if I multiply everything here by 2? Why? Everything will get scaled by the same factor, right? So if I multiply anything by a constant factor, in this case, it will not change. I can make it minus 5, 0, 5. As long as the relative scaling between them remains the same, if I multiply by this by a constant factor, it is not going to change. Once you reach, so your goal is to go from the start and reach towards the end. Once you reach the end, that's when the trace back begins. What is a trace back, right? So the trace back is connecting the dots looking behind in time or looking back in time. While moving forward, you're only looking for where exactly I came from. Once you reach here, which is your end, you try to figure out where did I come from, and you keep tracking back the path. And that's what gives you these two possible alignments in this particular case. We don't have to repeat doing that entire exercise because we did it in the last class. And hopefully by now you know what the calculations are. And hopefully what was not probably as clear maybe last time, and you can please feel free to correct me this time as well, is um, how the traceback happens in global alignment is slightly different from how it happens in the local alignment. In my global alignment, I have to go all the way to the end. I really, really want the two sequences to align end to end. So I will only start my traceback from here. What is this traceback? It's just reversing the arrow direction for each optimal path that you took. And there are two possible alignments in this case, because when I do the traceback, I see when I reach here, I see that I could have either this value could have either come from here, or I could get the same value from here. And that's why there are two possible alignments. Once you do the traceback, you should highlight whatever the traceback looks like. and that in this case gives you two possible optimal alignments. These are optimal, but these are two possibilities that happen. Is that part clear? Any questions or anything that is not clear, or the motivation is not clear? Anything that's not clear? How do we trace back, right? So we did all the calculations, right? We did all the calculations in the process of coming. So if when I was here, I would have figured out that did I come from here, 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 or and you would fill the entire matrix like that. Okay, but you will only keep arrows for you will not. So for each node, there are three possible arrows, but you don't want to keep all three because you only keep the one which is going to give you the maximum. That is what this looks like. Okay, so here, for example, I could have come from either this or this. That's why I'm going to keep two. But in most other cases, you're only going to get one. It again depends on what your scoring function is and what your alignment looks like. Once you reach towards the end, that means your alignment is done. So I start from here. This is a one score. I see, okay, this is the node. What is the arrow? This arrow is in this direction. I'm going to flip it. That means this is go diagonal, but go diagonal in the other direction. I reached here. Okay. When I reached here, I figured out that when I was moving, I keep track of where I came from. So I have access to this and I have access to this. I will see that, okay, this particular node has two arrows incoming, reverse them. So I reverse it and I can go to 1, or I can go to 0, and I can keep doing this step till I reach towards the end. And that path that you're going to keep track of, that is your alignment path. Okay. So I haven't purposely given you the pseudocode in this case, but once you do your assignment, the pseudocode isn't there, and you will see that how it's very straightforward. It's very simple thing to code actually. It's literally moving over matrices. That's what it is. What you have to be slightly careful about is how you do the trace back. Like you have to make sure that when you're keeping this, you're keeping. In most cases, you will not be able to handle unless you are very careful. Your alignment normal code will only give you either one of these paths. It won't give you both. You have to do something special to do that. But for this assignment, it's okay even if you give me one possible optimal alignment. Is that clear? There are only two, but it could have been three as well. It's possible that for certain scoring function, I could get -1 from all these three positions, and in this case, I will have three possible alignments. Okay, clear? Any doubts? Anything that's not clear? Please ask right now, because I'm happy to spend even one more few more minutes talking about this. Any questions? Yeah. So we'll talk about the local alignment in just a bit. But any other questions about global alignment? Yeah. Yeah. Hmm. Hmm. The reason that's a good question. So why are both the optimal alignments, right? Because the optimal alignment, when you did the optimal alignment, what you're optimizing for is the score that you get for each base at that particular location, having considered what you got previously, right? So what you care about is the score, the final score that you get. So if I take, for example, if I take when I made this decision, in this case, the only possible path that I could have come from is here, right? So all I care about at that particular step is what is the most optimal path till that location. I don't care about like what's going to happen after me, or what's going to happen, what happened before me. What happened before me is in some sense already implied in this end, because that's how you get your score. But you don't care about what is going to happen in the end. Once you reach the end, you try to figure out what are the possible paths. So you don't care about the total sum of the scores that you get. You don't actually care about overall path length, because in this case, both are going to be equivalent, right? How? Because once you do these matches here and you calculate the score, they will give you the same final score. And that's why the absolute you don't have to keep track of the entire sum till that particular location. That is already implied in when you're making that particular choice, right? Because at each step you're picking out the best possible. Any other questions? Okay. So why the local, so global alignment already does it, right? Like, why should we care about local alignment at all? And when I chose this example, when I did not choose this example, but I should have started and maybe then done a bit more biological interpretation. I had two different genes, but it wasn't as apparent as to what I wanted to actually motivate. So this is a one protein taken from different organisms, right? So this is fish and yeast, this is man and mouse, this is BRCA1. Now these are kind of different genes in some sense, but what I want you to focus on are these small subunits. So while proteins are one protein of one gene, let's say BRCA1, BRCA1 is a very famous gene for why? Breast cancer, right? As simple as that. It got its name from the discovery that was made in 1990s or 80s. So BRCA1 will make one kind of protein, and let's say I have another gene called GATA, right? So GATA is also GATA is another gene, which is a special gene called transcription factor. While these are different proteins, when you actually look at their sequences, proteins have something called subunits. So while this entire thing is one protein, and they are going to be different between different genes, it's possible that they share the same substructure, or let me call it subprotein. I can invent some terms for this class, right? Or I'll just call it some family. So within each protein, there are certain domains. Well, actually, that's the right name which I was looking for. There are certain domains within each protein that are very similar, because while you have only 20 amino acids, so maybe certain code of those amino acids are going to result in some structure. Proteins are very special in the sense because once we know the sequence, we kind of know what the structure it is going to be, right? Because, for example, that's what AlphaFold does. If you are familiar with AlphaFold, it goes directly from protein sequence to the protein structure. The reason that algorithm works is it seems that the sequence tells you, for example, if this sequence is MLM and let's say this is MLK, it will tell me how the protein is going to fold. Is it going to fold like this? And I'm very bad at drawing, but or is it going to make a very simple structure like this? What can happen is, while the entire protein sequence could be different, but these small structures could be still similar across different families of proteins. So if I have a new protein, even though, or if I have a new gene sequence, it's quite possible that that particular protein sequence will not fully align with all the other proteins in my data. But let's say I want to find out what are within this particular, you're still staying in the same organism. So I figured out, I did an experiment on myself. I figured out the protein sequence of all different proteins in my body, and of 20,000, 19,999 are what is already known. The 20,000th one that I got seems to be novel. Okay, it's novel. Nobody has studied it. I don't know what I do blast search. I don't find anything, right? Or let's not go to the blast search, but you try to find everything in other organisms and so on. There is nothing like which looks like this. Then what you would be interested in is, okay, there is a protein sequence. It must have certain function, and that function comes because of these domains. Are there other proteins in my body which share some domain sequence with the sequence that I have got? And that is the problem of local alignment, where you don't care about the entire thing, entire string to match. You care about that are there certain subunits, contiguous subunits, which maximize or which can align despite the entire sequence not being able to align. Are there things that if I come up with an algorithm, can it align? Now think about this problem. It's a much harder problem, very hard problem. Why? because it is combinatorial. Nobody has told you what are these boundaries. In this case, I'm drawing these boundaries, but in the real world, nobody will ever tell you what are these boundaries, right? It's very hard because they also don't know. So you not only have to tell you, you don't have to only tell me what is the correct alignment. So for example, in this case, one possible alignment, let me use colors. So this is two boxes. I'm going to call it P1 and P2. And then I have another box of protein, which is P1 dash, and maybe then there is P2 dash, and then there is P3 dash. Nobody has told you this, right? But the hope is that something has to be conserved. Like there has to be something functional. What is that functional? And what I want to finally give you is, I want you to tell me within this sequence, what is that position at which P1 starts, likely, and ends? If it does at all. What is the position at which P2 starts and P2 ends, if at all? What is the position at which P3 starts and ends? I'm not caring about things in between because maybe they're not as important, right? Like they're not, maybe they are very special to that particular genome sequence or that particular gene sequence. Is that problem clear? Why local alignment matters and why we care about local alignment? So it's a different problem. Problem is of looking for smaller sequences or subunits of a certain sequence which can be aligned despite the entire sequence not being very, very similar. Okay? So in order to solve that, we do two different innovations, right? So Smith and Waterman algorithm does two different innovations. Why does that innovation work is very, very important, right? So remember, once we make this choice that I don't care about things not aligning, actually, I don't care about it. What I care about is there certain subsequence which does align with good quality, overall good quality, and that can be ensured. So the first thing that you do is the gap penalties are zero. The reason gap penalties are zero is because I don't care if you get gaps. It's okay. I know that in this case, when I try to put them together, there will be gaps. Of course, there'll be gaps, right? Like this, I know, for example, will not align. And it's okay. If it doesn't align, just leave it. It's okay. Just take the zero path. Don't worry about, in your usual case, if you were getting all dashes, you will start worrying because your score is blowing up. -1, -2, -3, -4, -5, -6. But Smith and Waterman are telling you, don't panic. It's fine. It's going to happen, right? Like we know that this sequence you cannot find anywhere. So don't worry about it. Just make it zero. Okay? That's why that gap penalty is zero. Yes. Yeah, the good quality alignment is once you figure out the alignment, your overall score is as positive as possible overall, right? Zero score is not good, basically. Anything positive is good overall. And you're looking for what is that contiguous sequence in which you get a positive score. Okay? Why zero matters here, is that clear? I don't care about things not aligning. They will not align. It's okay. But if there are certain matches, then you should be able to sort of see them. And how do you see them? Since we want things to align in a nice fashion, we now care about higher scores. We now care about what are the nodes where we get maximum or very, very high score. Okay. So the Smith-Waterman, the only distinction that Smith and Waterman are doing in this case, sorry, I forgot to put the scoring matrix. The scoring matrix is not very different, but it is max. Max of this can be zero. Smith and Waterman can never get negative scores because you don't care about alignments. So it's max of Sij. So intuitively, what you really want, right? You actually want this to happen all the time. In the case of Smith-Waterman, you want really good alignment, really, really good stuff, right? Really good things which have maximum overlap at the cost of not being able to align the other stuff. So you want this to be, ideally you want this to hold all the time, but this is life and not everything is exact. So it's okay to allow for deletions if you really have to, because sometimes, sorry, I'm writing it very poorly. It's all, you already have access to it. So chalk has kind of given away. So while indels are allowed within those sequences, but what you really are hoping for is I get a perfect match, right? So you have multiple, you have written documents all through your 4, 5, 6, 10 years of career, whatever you have, and you're looking for certain keywords. It's a keyword search that you're doing in your thesis or in your report. And what you're hoping for is you get very, very good hits of those keywords, right? That's what local alignment is trying to do. Big document being your entire gene sequence, and small keywords that you're looking for and you want them really, really quick. Okay? You don't care about that if I search for THE and I have, let's say, okay, this requires innovation. So let's say random word. Okay. What I'm looking for is, if it doesn't happen to be there, right? If it doesn't happen to be there, I don't have to force things to align and come up with maybe this alignment. What I really wanted to show up is there a the pattern somewhere coming up, which in this case was missing. But if this is your entire thesis, for example, you prefer getting a subunit to match perfectly rather than having to figure out what is the right alignment. In this case, if you were to begin, it's possible that you would have gotten this alignment, which you don't want. You don't prefer this, because this is one good local fit, tight fit here, which you prefer. Okay, that's the difference between global and local alignment. Clear? Any questions? Now comes the important part, which I very quickly skipped over last time. How do you do the trace back here? You want good quality stuff. Good quality stuff here refers to higher overall score till that particular path. So to begin that, to do that, you look for where in your graph do you get the maximum possible score. Because your alignment can begin anywhere. It's neither, it doesn't have to necessarily begin here. It can begin here, 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 here. But it has to, of course, end somewhere, no? So you figure out that, okay, 5 seems to be the biggest where it seems to be ending. This is the biggest value that you get in this matrix. Your trace back begins from there. Okay. Now, once you figure out this, you just do a simple trace back and you stop if you start getting indels, which means that all of your score goes to 0. So you do a trace back and that gives you a local alignment, but you still figure out that there are, in some of the nodes, there are possibilities of getting, see here again, there is some possibility of getting 1 both coming from 3 and coming from 2, because there is an indel, and that's okay. You're getting it, but you stop at that location, right? Remember, this is a longer sequence. I'm not showing the rest of the sequence here. I'm just showing you that ACG does local alignment with ATTG, which is a subsequence of your longer sequence that you began with. Does that make sense? Any questions? You will stop once you get a 0, because the 0 value in this case represents your start of your indels, like start of dash, 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 dash. So you stop here, right? You stop at the location where you get a score. This is the beginning of your alignment. So once from any particular location, if you have to make a jump to 0, that means you came from a dash, dash. That's how you would get a 0 in this case, right? The 0 is max of 0 plus all other scores. There is no negative stuff. The only way to get 0 is through those dashes. So you're saying that I started from this where I got the highest score. I went back, went back, went back, went back. But here it should stop. I should stop here, right? Because after that there are dashes, which I don't like, because it's a local alignment. Any questions? So once so so there is once how you fill in terms of your zero, that is the only difference between local and global alignment, right? So the traceback is not any different. How to initiate the traceback, that is different, right? So you initiated it here and not here, even though this is the end. Why? Because this is high. So after this, you're going to suffer, right? Your score have dropped, which you don't want. You want really good quality stuff. It's okay if it is two bases. I don't care, and you'll see. We'll see an example in just a bit. But I want really good stuff. Yeah, yeah. Okay. So why should we prefer three? Yeah. But the moment you figure out how you reach there, right? Like once you had reached three, after this particular path, you could have whatever the next step that you're going to take. It's going to give you the same cost even if you have taken a different path, right? So that's why it doesn't matter when you're tracing back. This score does not matter. While coming down is what it matters because there are different possible alignments. So till what it is showing you overall there is how many paths are there to reach till this particular base, and you can either do AC dash or you can do A dash C. That's basically analogous, and you can see this from the sequence also, right? Okay. Any other questions? Okay, now comes the fun part, which is we live in an interesting era, right? Like, sometimes you have so many ideas. My first website that I coded was in 2012. There used to be language, I think it still exists. It's called PHP, and your first websites used to be in PHP. And if you made a login website, you'll be very happy that I have a login website where you can log in, you can create users. Everybody would make a library management system to begin with. If you go and search some of the CSE websites, old websites, you'll find a lot of stuff like that. But now we live in very interesting eras. If you have ideas, uh, it's very easy to prototype things. So this is my attempt at doing vibe coding, which it's wonderful actually. It is extremely powerful. So I've always suffered with this problem, right? Like how to teach this, how to make it intuitive, how to sort of get a flavor. And I'm still not there yet. Like I think it's still not there, but it's a good interface, I feel. And you can tell me whether it works or not, because you can use this to test your assignment, and hopefully if you can find out bugs, that's also great. But what this interface allows you to do is basically I can run both global and local alignments side by side, and I should be able to see overall what is the difference between a local alignment and a global alignment, right? So in this case, for example, here I am forced to do end-to-end alignment. So I really have to force these dashes. This is a forced example, of course. Otherwise, your local alignment and global alignment are going to be the same. We can take any other example. I'm going to just maybe cut this down and see, well, in this case also it's going to be the same. But in this case, you will see that if I were to force global alignment, it's literally wasting my effort trying to literally force this. Whereas I can see that the global alignment is actually just at ACG level. Okay. Any questions? You can also see how the traceback happens, for example, here. It also explains, and I think it's correct, is how did you reach each of this step? Like what are you doing at what step? What information is being taken, and how do you actually do the traceback, and why is it different between a local alignment and the global alignment? Okay. Any questions? Okay, then now the fun part of the course begins. So thank you for sticking around. It's been a slow ride, but now we ramp up a bit and we try to talk about some biology, some biochemistry. So we're changing some gears, but then bringing in algorithms and statistics in just a bit. So this lecture will focus on an interesting part. Yes. Hmm. This one? So that's what, right? So I stopped at ECG. In this case, it's a forced example because only 3 nucleotides. So I stop once I going to get a 0 after this. No, you stop when you get a 0 because it could have happened at any position. Okay, that's a good question. Let's go back. Let's go back to this example. It's interesting because it's very interesting. I can also tell you. So let's focus, right? Any other place where you would like to have started? Here, you're looking for maximum stuff. Where is the maximum stuff? Here. Any other place where you would like to start this alignment? What is happening here? It's pretty large. Still, 4 is pretty large. So this is also a suboptimal alignment possible. I could have started here, which means that I would align my G to T, sorry, my G to A, and that would also give me one possibility of starting and just simply ending, one base alignment. So you stop once you get a 0. I would have started here because it looks very high. I would have gone back and I would have like, okay, that done there. That is the only possible start point that you get. Okay, so you start at the maximum possible location, you end once you get a 0. Okay. Okay, any other questions? Okay, so now fun part starts, and I asked you to remember where did I come from, right? So where did I come from is an interesting question because I asked that question to myself multiple times, and this has been studied in Indian context in different papers, right? So broadly, India is made up of different. You can divide India in every possible way in terms of its axis. You can start from states, you can start from districts, you can start from genetics, you can start from which languages we speak. And in this case, this is reflective of the languages or the language lineages that we have. This is taken from a 2012 paper. We are either Indo-Europeans, we are Austro-Asiatic, we are Dravidians, we are Tibeto-Burmans, or we are Andamanese. This only tells you half of the story because where you came from is a very tricky question, right? So while languages only explain one half of it, the reason I was interested is I really wanted to know what languages my ancestors spoke, even though I had some ideas. And one way to do this is to, of course, study those languages and flow. Where did we come from? Iran? Did we come from, or did everything originate from India? Which is what one group of ideology in India believes, which is totally incorrect. But anyway, so other way to do this is to actually do what's called genomic sequencing in one way or the other way, right? What you're looking for is molecules, you're looking for DNA sequences, and then you're trying to essentially find out which other population of people where you know where they came from does your DNA have matches with. You will not take your entire DNA, you will take certain segments of your DNA and try to do a match. And this has all become possible by this technology called genome sequencing and certain precursors of that particular technology. So this is taken from my genetic profile testing done 11 years ago now, which told me that I'm Central Asian, Northern Indian, and Pakistani. All of that, some of that is true. I have a North Indian and Pakistani origin, Bihar, which is correct. I could also have share some ancestry with Bengali and Northeast Indians, but I'm broadly Central Asian, North Indian, and Pakistani. Right. So this is done by this company called 23andMe, which almost went bankrupt, and I think it's now trying to come up and get revived and so on. But what that company tries to do, and there are a lot of companies like that, is look at you basically get a kit, and in that kit you spit, and that spit is shipped, and from that spit you figure out your DNA, and from that DNA you figure out all this information. And this is only part of the story. It also tells you what kind of mutations you have, which predispose you to certain things, diabetes. One of the interesting thing using those times used to be this ability to flush alcohol. So there are certain Asians who are not able to flush alcohol, and when they drink alcohol, their cheeks become very, very red. So you can figure out from a genetic sequence whether you are predisposed to it, whether you will be able to flush alcohol or you'll not be able to flush alcohol. Despite that, please don't drink alcohol, whether you have the ability to flush or not. Okay. Okay. So this has become possible because of course, because of genome sequencing. And this is only one question that, of course, this answers. But the power of being able to sequence something or being able to study something at molecular is phenomenal, right? So our goal is to characterize the molecular map of the associated phenotype. What is a phenotype? A phenotype is anything that you can somehow measure or observe. The reason I behave something like this is something to do with the molecules that are shaping me, and I would really want to characterize this at the molecular level. The modern approach to do this is to this machine. This machine is called a sequencing machine. You throw off your DNA on this, and it spits out those sequences of A, C, T, and G. While this journey is a journey of biochemistry and biology, the data that comes out is purely handled using statistical computer science based approaches. And that's what we're going to talk about today. About before we go there, what is it that happens there? So the entry point for this is this interesting reaction by Kary Mullis, an eccentric person who wrote a book also on his discovery. And I would really encourage you to go and read about how he discovered. Apparently, he was driving on what's called the Pacific Coast Highway, PCH, and got this idea that you can make copies of DNA by using a very simple idea. What is that idea? The idea is that I want to make copies of my DNA. Why? Because the amount of DNA that I have in my body is very, very small, or in my cell is very, very small. So if I want to characterize the molecular makeup, I want a technology which can measure my DNA. But that DNA is so small that by the time I capture it, it's going to just be so small that I will not be able to do any analysis with it. So he thought, okay, we should be able to somehow duplicate it. Before we do anything with it, we should be able to make copies of it. And your body already does it. Your body has a mechanism where it's able to make copy, because cells keep dying and cell has to replicate before that. And this is kind of the principle that you want to emulate, but outside the human body. So in PCR, your goal is that this is your target DNA. You want to make more copies of it. So in order to do this, you start by just heating this DNA, or what is called the denaturing reaction. You heat it to 95 degrees Celsius. The strands, which is double strand, will break. You will get single strands of DNA. You're going to now cool this to 50 degrees Celsius. You will anneal what are called primers, and that's where the innovation happened. Primers are nothing but special sequences which will reverse complement to your sequence of interest. So there is some circularity here. I want to make copies of my DNA, but I need to know something about that DNA, because I would like to, for whatever sequence I know from this DNA, I would like the primers to be designed against that particular sequence. So these blue things that you're seeing here will will attach as primers. You will now add stuff. You will add what are called your nucleotides, your 4 nucleotides into this reaction chamber. And you will add something called the polymerase, which we had very briefly touched upon in the first class. And the polymerase will do its magic. It will figure out that there are these loose ends which it needs to complete. It has this inherent property that anything that it sees as incomplete DNA strand is going to complete it. So it's an enzyme. That enzyme will go and start this DNA synthesis. From these one copy of DNA, once it finishes one round, you would have already made 2 copies. And you keep repeating the cycle again and again, again and again, again and again, until you get the amount of DNA that you want. Each of the strand that you get, so this is called the first pass or the first cycle products. This is your original, gray is your original DNA, and the rest is reverse complementary country strand, which is made of entirely new sequences, right? These are what you added to the reaction chamber. You will do one more round, and now again these will split, and now again the synthesis will happen, because you again have the chambers with primers and you have it with your nucleotides. polymerase will again extend it, and it will keep happening. So from 1 you go to 2, from 2 you go to 4, from 4 you go to 16, from 16 you go to 16 square, and so on. So it's exponential amplification in that sense. Does that overall make sense? Why are we doing this? Because we want to make multiple such copies. So you start by DNA, you mix target DNA with your primers. You need 2 primer sequences, one which reads your forward strand, the other one which reads the negative strand. So here you need one primer which is reverse complementary to this part of the DNA, and the other one to make this other part of the DNA. What happens if you only use one? What happens if you don't use this primer? Thought question. No biology required, no logic required. If you understand this logic, you'll be able to answer it. So at the end of, let's say, 5 cycles, how many copies of DNA will I have? So in this case, in normal thing, I will have 2 to the power 5 copies of DNA. If I only use one strand of primer, how many copies will I have? Every time, yeah, sorry. Every time I'm adding only one copy of the primer, yes. Yeah. No. So you let no. Good question. Good question. No, you let the reaction you initiate it with one strand, one primer only, and then you let the reaction run as it is. Okay, what will it increase like? So quantitative numbers, 2 to the power 5 is what happens if with 2 primers. What happens with 1 primer? Interview question. Well, not for finance companies, but or analytics companies, but other interviews. To the power? Will it be exponential? Is the first question. It won't be exponential. It won't be exponential. I will encourage you to think about the number. It won't be exponential because at each step, right? So this is the only primer you're adding. So you can only be able to synthesize this strand. Think about what happens in the next cycle based on what is happening in this figure here. So the reason I'm thinking about these questions is because these are the questions I generally like to ask in the exams. Is you've studied the concept, right? I don't care about you remember this temperature or not. Nobody remembers. I don't remember. I don't care also. Only one strand. Okay, why? Think about it. Think about it. But anyway, the reason for asking that question was to give you a flavor of my exams are the midsim will have some theory, but all thought experiments. What happens if this happens? What happens if that happens? Right. So just that's why I focus on the concept, temperature and all. Don't worry about it. Basically. Okay. Right. So what is mechanistically what is happening that at 94 degrees Celsius, your hydrogen bonds of the double strand are broken. Your target DNA gets denatured, and TAC is the name of the polymerase. So the innovation here was actually anything will you find weird? DNA is denaturing, but nothing is happening to that enzyme. Why? Somebody said it. Okay. Thermostable, right? So it's a thermostable. So this is taken from what is it called? Thermophilic bacteria, which have a very stable, they have survived in very hot temperatures. So they have a special polymerase, which is called the TAC polymerase. It's thermostable, and that prevents the decay of the enzyme itself, right? Again, another thought question: why does it not? Right? So why it works? Should not work, right? If DNA is getting denatured, everything else should also get denatured, but it doesn't because that's how it is designed. So those are where that's where the innovation happens. You raise certain temperature to certain degrees because you want to match the physical conditions of where this was taken from, and it's apparently very stable and very efficient at 72 degrees Celsius. That's why you go through this cycle of denaturing and renaturing and increasing the temperature to 72 degrees Celsius. You have seen PCR in life, right? some version of PCR. We are all COVID. We have all seen COVID in our life. Does this recall? Can you recall what version of PCR you saw? What is RT there? Okay, reverse transcriptase. Any other options? Okay, that's a. So this part is just a repeat. This is taken from Wikipedia, which I would recommend. I've already talked about this, but let's talk about real-time PCR first. So in real-time PCR, the idea is when you get the products for normal PCR will take you that cycle, right? And that cycle can take up to how many hours does it take? You've done PCR? How many hours does it take? 2.5 hours for how many cycles? Okay, fine. So like few hours it takes, right? I don't want to wait that long. I would like to in real time be able to see that is my DNA of interest getting amplified, right? So it's also a diagnostic in the sense that if you know that this particular DNA sequence exists in your sample and you design your primers designed for that particular DNA sequence, that particular DNA will get amplified. That's what was being done very briefly in the case of your virus detection, right? You're able to detect that virus because you know the sequence of the DNA. You design primers against that particular virus, against those locations which you know will usually not get mutated, because viruses are prone to mutation. So that's why some of the kits in India were, and actually also in the world, were beginning to fail at some point, because if you have a new variant of the virus and your primers are designed against those areas where the virus mutates a lot, your detection kit will fail. But broadly, that's how the detection kit of at RT-PCR for any virus or any bacterial pathogen, for example, works. If I want to do this in real time, I have to do some more innovation. I have to be able to see this thing in real life rather than have to wait for 30 cycles to happen. So what you do is you take a reporter probe. This is your usual, which is called a probe, which is basically similar to your primer, but you have a fluorescent label attached to it. And this fluorescent label, it's a probe because it's circular in nature. Once it hybridizes to your target DNA, it's going to, if it is in this particular format, it quenches the signal. So it's fluorescent, which means it's going to emit certain kind of light signal. By default, it is quenched, which means that if they are next to each other, you'll not see anything light up. But the moment the amplification starts to happen, you would start to see more and more of this light. And that's what is done in a real-time PCR. Is this what is used for COVID? This is also a real-time, right? RT-PCR. Yeah, so what happens when you have the target DNA, this probe, right? This probe is otherwise stable in its loop kind of format. Once it sees the target DNA, like anything in life, it will try to find even more stable structure. And there is a denaturing step happening. So it will hybridize to the DNA if you have the DNA target corresponding to the correct primer that you designed this against. And once that happens, these two things fall apart. What was being quenched earlier in terms of its in terms of its fluorescent signal will not be quenched anymore. So you will start seeing emission of green signal in this case. And that your laser or any camera can read that this particular signal is coming again and again. It's lighting up as your PCR cycle goes. So you'll be able to measure without having to wait for 2.5 or 3 hours what is the overall content, how much emission do you see overall. Okay. Okay, so that's not RT-PCR though, right? So in RT-PCR, you are working with RNA to begin with, and RNA is hard. RNA is always hard because RNA decays very fast. DNA has a very stable shelf life. We can detect DNAs of dinosaurs. We can detect DNAs of Neanderthals. We can detect DNAs of ancient humans. But RNA very hard because it's, it requires very special conditions. So it's that special kit which requires a lot of massaging and a lot of stability for keeping it intact. So to do that, to work with at the RNA level, what you do is use a very simple idea, which is what a lot of viruses also use. Is you use another enzyme called reverse transcriptase, which will read the RNA, convert it into DNA, And then the rest of the process more or less is very similar to what you do in your PCR reaction. You're just converting this, and that's what you do in case of your virus, RNA viruses, in terms of how to detect them. Okay? Why are we talking about all this, right? The reason we're talking about all this is what I'm going to talk about next is very much reliant on you understanding why we need PCR. PCR is required because the amount of material is very, very small. The goal that I promised you this that, or we began this session with, is I want to figure out where I came from, and I'll not deliver on that goal yet. But before I do that, I have to understand that what does my DNA sequence look like. So my DNA sequence is 3 billion base pairs, and I told you this, but somebody would have measured this. In this case, I also want to know that what is each individual base of my 3 billion base pairs, and somebody has to come up with a technology to measure it. In old days, what they would do is they would take your entire DNA, they would fragment it somewhere and try to figure out what is that particular sequence. What is the biochemistry? How does that work? That I'm going to talk about in just a bit. But any ideas that if you have to, if I gave you access to a black box technology to sequence, let's say, part of this DNA, right? No technology I can tell you right now exists which can read this entire billion base pair molecule at once. But there are technologies which can read them in parts. So if I gave you access to that black box technology, how would you put this together? It's very difficult to sequence all the 9 billion base pairs, right? So PCR also starts to, or any other polymerase will have will have what's called the fidelity problem in most cases, which will prevent it from amplifying anymore or creating copies of this particular DNA anymore. If you had a mechanism which you can read each base, right? The PCR is not about each base; it's just about amplifying the DNA. But let's say I give you a black box technology which can read, which can tell you what are these individual bases. But let's say it only tells you 50,000 bases at a time. How would you put this together? How would you put the 3 billion bases together at once? Yeah, simple idea, right? Overlapping. That's the engineering approach, right? So this is exactly what the science of assembling the human genome looked like. So this project was conceived in 1990s and was delivered in 2003 by two individuals or two entities, excuse me, one being Craig Venter. Craig Venter came from the private side, had this company called, forgetting the name of the company now. I think it's Celera, but we'll find out in the next slide. And then there was another public project which was funded by the US. Both of these were funded by the US government, and you'll see three distinguished individuals come up in the next slide. But the basic idea on which these projects worked is we wanted to determine what is the human genome sequence, what is the human DNA sequence. They took this entire DNA sequence, we chopped them off, we sequenced very small parts, and like a jigsaw puzzle, you try to put them together. The biology problem stops once you get this sequence. The computer science and the statistics problem begins when you're given these sequences. How to put it together. It looks very easy, but a very, very hard problem. Still an open problem in some extent. It's still not a solved problem. How to actually put it together. Okay, you did not get what the problem is. So we have 3 billion base pairs. Okay, very long stretch, right? I want to figure out what is the individual sequence of this. What is the full sequence? Okay. Now, no technology can measure this in entirety at once. Okay. But there are technologies which can tell you that this particular sequence is whatever it is, ATCAG. If I want to now, with using that technology, I want to put it together, how would you do it? Put the entire, this is your puzzle, okay? These are your pieces of the puzzle that you can, that I gave you access to. How would you put it together? Got it? Everyone with me? Yeah, please stop me if my if the motivation is not clear, because it actually defeats the purpose. Then you will not be able to relate to the problem. Okay, clear? Right. So one way to do this is you can sequence short fragments, right? But I actually, to begin with, I don't know what am I going to, like, where am I? Like, the technology does not have access to specific locations. You know, it knows that if you give it something which is, let's say, 100 base pairs, it will tell you what is this 100 base pairs. But there is no technology which will first read this, then read this, then read this, and keep emitting out. It doesn't exist. So what do? You chop this off, right? So you take your entire DNA, you chop this off, and then you sequence those 100 base pair sequences. Can you still put it together? So for example, sometimes the technology will see this, sometimes it will see this, it will see this, it will see this. Let's say you have this sequence. How will you put it together? How will, from this technology also, how will you make this a contiguous sequence? Is that problem clear? Any ideas? Okay, we'll try to find overlaps. So we will see this in the next class, but we still have to still have some biochemistry to cover. So this Human Genome Project, we now know what my billion base pairs look like, thanks to the Human Genome Project. It was delivered much ahead, one of those projects which was delivered much ahead in time, because there was this competition from that person that you see on the left, J. Craig Venter. And the person on the right is Francis Collins. The person, of course, in the middle does not require any introduction for a variety of reasons. Hopefully you know him. But this was delivered in 2003 or 2004-ish, around for 5 billion US dollars. But one thing I want to always highlight is the contribution of this individual called Jim Kent. He was then a graduate student. He was like, just like a PhD student, like some of you. And through spending hours and hours writing algorithms, knowing that somebody else was also doing the same thing, um, he developed a lot of tools which are still used in the field for putting this data together, for what's called assembling the genome, right? So it's in what's called the genome assembly problem. You're trying to assemble from short fragments what the long fragment would look like. I would really encourage reading about him. He's still a very active contributor to a lot of bioinformatics algorithms. How much does it cost, right? So 5 billion was what it costed that time, or what it used to cost. And this drop has been what beats the Moore's law. What is Moore's law? Every 2 years, the number of transistors that you have in your chips will double. But right now, we are very close to getting a $200 genome, and this is, it's like half baked promise. In India, we unfortunately still cannot do it. We're getting close, but the challenge of delivering a $200 genome still remains. There are companies which say that if you give them your DNA, they will be able to give you the entire thing in $200. And it's close to reality in the US, but still not something which we can see in India. Anyway, but how it got delivered and how we reached there, the next few slides will very quickly talk about that problem. This problem of being able to put together DNA is very, very old. It has been something which people have been thinking about for a long while. And that's why the technology to measure, right, the black box that I was telling you how to measure it, went through different generations. There's first generation, which is called Sanger sequencing. The second generation, then came the microarrays, which I don't put into any this thing, and I feel nobody should use them. But third one is second generation sequencing. And finally the third generation, which is starting from 2000, which is what we are at right now. So what is the concept of being able to determine these sequences? What is the problem? The problem is I want a black box algorithm or a mechanism through which I can do an individual read of what my base is. So if I give you this sequence, right, I gave you the sequence, but I don't know, right? I did not know before that I gave you the sequence. Can you tell me what are these individual bases? Sanger sequencing and all these four sequence technologies that I talked about talk about or are solutions to that problem. The idea behind Sanger sequencing is very, very simple. What we have in your usual bases, there are 4 bases, A, C, T, and G, and broadly they look like something like this, right? So this is your base. It is attached to another base by what's called a phosphodiester bond. What you do is in this particular base sequence, there is a hydrogen at the 3rd prime or at the 3 prime location of that ring. Again, the chemistry of it doesn't matter, just focus on the logic. What are we trying to do? We are trying to determine, given a particular sequence, what is the corresponding, given a particular DNA sequence, which I don't know the sequence of, what is the corresponding sequence in it? Is it TTT, is it AAA, or whatever? Okay. So to do this, what we do is we start with a DNA strand. We start with a reaction chamber, and in that reaction chamber, we put certain special nucleotides. These are called dideoxy, okay, DDNTPs. The other ones are your DNTPs, deoxynucleotide nucleotides. In this case, these are dideoxy. These are dideoxy because the 3 prime location of where your usual base, right? So what we are doing is instead of an A, you have something called a special thing called A star that you have synthesized. Okay. Now, what is special about this A star is what there was an H, there was an OH here that is being replaced with an H. Okay? And that's what makes it dideoxy. Now, the reason this change is important is because remember that DNA will form chain, right? Like this is all connected through a chain. It's not just any random sequence. To form that chain, you require the presence of OH, because this is, if you have studied chemistry, if I have to okay, so this is your OH. What do you think if I, this is from base A, and let's say this is your N, this is your C, which also has an OH. If they have to form a bond, what should get released? Yeah, right. As simple as that, right? So that OH is the one which gets cleaved essentially, and that's how it forms bond with the other thing. So this allows it to form key forming bond, but if you change it to H, it can no longer elongate. So if I incorporate a DDNTP in my chain here, it will stop. If I incorporate it here, this will not be extended anymore. That is the key idea that Sanger sequencing relies on. So what you do is you start your reaction. In this reaction, you start with your DNA of interest. You take DDNTPs along with DNTPs. So you have the normal bases of A, but you also have the, let's call the abnormal bases of A. That abnormal base of A, C, T, or G, if incorporated, will prevent your DNA sequence from getting extended. Okay. So let's assume this is your DNA sequence of interest. You added 90% of this was normal A, C, T, and G, and 10% of these are DDNTPs, dideoxy ones. This is your chamber, right? This is your DNA sequence. I add polymerase. It will start to read through, it will start making reverse complementary part of it. I keep going, and if it is blue, it means I'm incorporating the normal one. And if I'm incorporating the normal one, of course, it will keep on extending. The moment I incorporate abnormal one, I cannot go any further. So let's say this happens once. Okay? Now, since I've added 10% of other stuff, there is a chance that at any point here, I can get the abnormal one can get incorporated, which is the DDNTP here. So again, I'm going. I'm it's extending, but at some point, that abnormal A DDNTP gets incorporated, and this cannot further extend because it doesn't have an OH site. It only has an H, which does not allow any base pairing to happen or any other pairing with other DNA to happen, and that stops. Same time, this can happen anywhere in this particular sequence. Okay. Because of this, you get a ladder, right? you will get fragments. Your original fragment length is this, but because of this kind of special reaction that you're doing, you will get lengths of different lengths, DNAs of different lengths, which you run through this gel. Now, this gel is an interesting gel, and the simple idea that gel uses is DNA has a negative charge, and longer length will have if you add positive thing here and if you add negative thing here, things will try to move towards the positive side, and what is longer will have higher weight, so it will move lesser, and so on, right? So that's the broad idea. We don't have to cover gels, but what you can do is, this is the pattern that you're going to see, right? So this is T. The reason it's here is because it's a very small sequence, and the reason this particular sequence is here is it's because it's one of the longer sequences. As you run this through on the gel, these are DD NTPs. They actually also, you can think of this as having some sort of a label that you can read, a fluorescent label, just to imagine. You can follow this chain to figure out what happened first. So I got a T, then I got a C, then I got a G, and so on. And that's how you get your entire DNA sequence. Does that make sense, intuitive sense? It's a reading mechanism, right? You're designing a reader for DNA sequence by leveraging the properties of the base and just taking the engineering approach, changing it, and being able to figure this out. Okay? Any questions? So this is where the smallest sequences are, right? This is bottom is where the smallest sequences are. A sequence of particular length will have a particular footprint. Let's call it footprint, which is basically it will have its own mark. So here I read and I see that it emits a particular signal. It belongs to this T thing. In the next slide, I will show you what exactly is done. But you can think of this that it is emitting a red light, for example. T is emitting a red light. C is emitting this C is going to emit a green light. This is going to emit a blue light, and this is going to emit a yellow light, for example. Okay. When I read this from this gel, this is the smallest sequence. So I see that a red signal is being emitted here. So it's the smallest and a red signal. So I know that it is T. I go up here, okay, which is the next longer sequence that I have on my gel. And I see that it is now emitting a green, which is corresponding to C, for example. So I get T, C, and I keep going one base at a time overall along this trajectory, because this trajectory is dependent on what is the length of your DNA, essentially. So each length will correspond to one spot on this channel. We do get abrupt things, but it does work very, very well. You have to just fine tune the 90%, 10% ratio overall. That if you get mostly that right, you don't get many abrupt emissions. Because even if you have, like, while this is one example, right, like you'll have population of these, right? And since all of them are being equally added, that just by law of large numbers, your final proportions that you see will reflect the overall content of what this DNA looks like. Any other questions or any other thing that's not clear? Yeah, so that's a good question, right? This is slightly forced example because, of course, you will see normal base pair incorporations as well. But what you're reading is you're reading only the signal which is being emitted by these special base pairs. So even if there is an incorporation, right? Or are you saying that 2DD NTP is being incorporated? That's possible. Yes. Yes, that's possible. Which one? Okay, here. Why? How would it get CTT? Because this is going to be complementary to what you have here. I see. Okay. Let me show the other version. Let's look at this. This will hopefully make it much more clear in terms of what I think I understand your question. So overall, right, the reason you will get a lot of these kind of fragments overall, right? And the moment you have a length, you know till what point you have been able to replicate. That is going to get fixed, right? So if this length increases by 1, that would mean that I've incorporated a normal A already, which I know the corresponding sequence for, because I would have otherwise terminated it there, right? So that information by that time is already redundant. Does that make sense? So that is how you're able to sort of read this signal, right? So in this case, if I know DDT and if I know that this is one length longer than the sequence, then I have to go in this direction to read it through, right? So that's why that stop gap will option will not happen, because you're moving one base pair at a time. Okay. Any other questions? So this is your red, blue, green, and orange. You put this in the reaction along with your DNA, along with your polymerase, along with your normal NTPs. This gray stuff is when it's normal, but if it incorporates a non-normal stuff, it will stop there. It will have a particular length, and this keeps on happening until, of course, you get a very, very small length. At each point, you will emit a signal. You can measure the length of this thing, right? And you can figure out from this kind of a chart what does the overall sequence look like. So if I read the signal, right, like this particular signal first is C, it's the first signal that I get there for that particular sequence, and so on. So I can read it on my chromatogram, like imagogram, what this look like. It's not a chromatogram. Okay. Any other questions? If you understand this, the rest of the stuff will follow very, very easily. However, why we stop there, right? Like it can only go up to 1000 base pairs. It is accurate only for, it's very slow. So if you want to sequence, you will have to spend 1 day sequencing only 1000 bases. So in your lifetime, or in my lifetime, I will never be able to finish, in my remaining lifetime, I'll never be able to finish the sequence of my own DNA. It is also not accurate, like its accuracy starts to fall off. I think that probably also answers your question. Till 700 base pairs, it is okay, but if you go above it, it starts to have those random issues. Okay. So I think, how much time do we have? 5 minutes, right? 2 minutes. Okay, thank you. 2 minutes, this is good for 2 minutes. Microarrays, 2 minutes is good. So the idea behind microarrays is one step up. At this point, we have a technology which can read it, but it's very, very slow. But by this time, when I'm in 1980s, I have already known a lot about my DNA sequences, because people have cloned multiple times using Sanger sequencing. So I know ballpark what primers should I use, right? Like I have access to what are the different sequences that shape up a different gene. In microarrays, the idea behind microarrays is you want to sort of see whether a particular sequence exists or not. The way this is done is you take a chip surface, and before even you start the experiment, so you're a commercial company, what you do, you manufacture microarrays. What are those microarrays? You take a glass slide, and on those glass slides you make holes, and in those holes you stick what are called primer sequences, or probes, in other words. Now these probes have some random A, T, C, and G, or whatever, some special sequences, and you know that this sequence only exists in gene 1 or gene 2 or gene 3 in let's say human DNA, because you've studied this, right? Somebody else has already characterized part of it. So by implanting them, you put them so that now when you actually put your DNA, I will be able to make copies if it reverse complements with that particular sequence that I have. So if I put my DNA, if the company designed probes against 5,000 human genes, I can only quantify whether I have those 5,000 genes present or not in my sample. Does that idea make sense? You're essentially trying to quantify, in some words, how much of a particular, this was not developed for studying DNA so much. This was developed for studying RNA, which we will cover in a couple of slides, a couple of lectures down the line. But the overall idea here is very simple, that rather than doing a PCR, you are taking the different probes which are fixed, which are your primers. You're putting your DNA or RNA, which will reverse will form reverse complement. It will hybridize. You will do all those reactions, And at each point, again, if it hybridizes, it will emit a signal, which is what you're going to capture. So if I have a particular location, if I have 5,000 spots on my glass slide, these 5,000 spots correspond to 5,000 genes. If all of them light up red, it's great. I have all those 5,000 genes. If I had cancer, and again I put it, this will, some of them will not turn. I could, rather than red, I could now put a green, and I would then overlay them over one each other. And if you overlay those two, so I'll just take 30 more seconds and finish. If you overlaid two different microarrays, right? So this is R and this is yellow, for example. I forget what are the primary colors. How do you get green? How do you get green? Red plus yellow is green? Okay, there are a lot of people like me. Which gives, what gives green? Should be something, right? What is it? Okay, fine. Let's make our own. It's fine. We can make our own. That's fine. Blue plus red plus blue is green. Okay, I think this is correct. Oh, is it? Okay, so blue plus yellow is green. Fine. Okay. So I added in one case I added red, and I overlay this with another one in which I have blue. If they overlay over each other, that means I have both of them, and they will make green. Hopefully, they'll make green, right? Yeah. So they'll make green. In which case, everything will green. I don't have cancer. But if some of them are only showing red or blue, it means there's something wrong. That either what should be present is not present in my sample, or what should not be present is now present. So this technology is used to detect diseases in some fashion, that you can take this sample from two different individuals, figure out what is common between them and what is different between them. You can only, however, do it for, the reason you cannot use it in a high throughput fashion is you can only capture things which are already known, and only a subset of them. I cannot do this for all 20,000 proteins. I will maybe able to do it for 5,000 or 10,000. I cannot do untargeted discovery, in other words, using a microarray. I think it's a good stopping point. We start next lecture with second generation sequencing. There is one question in your assignment which uses material from this lecture, which is on PCR, but that's all. When you get your next assignment, we'll cover the rest of the stuff from this lecture. Thank you.